We are very happy to welcome you on a new BrainMap seminar. And I just want to say that this seminar series is co-sponsored by P49 funded Center for Mesoscale Mapping housed in the Martinos Center. And today is a really great pleasure for me to invite Dr. Pantelis Lioumis to give us a talk about brain mapping. And I just want to say that Pantelis was the person who was teaching me how to do TMS more than 10 years ago when I was just starting. And I'm extremely grateful for him for this. And he's proceeding to do really great job studying brain circuits using TMS, EEG, MEG. And Pantelis, Dr. Lioumis is working at the Aalto University and Helsinki University. He's a adjunct professor, he will talk about TMS and TMS EEG. And I think, Pantelis, I think you can introduce yourself even better than I can do. So Pantelis is really one of the, in my understanding, pioneers of the speech mapping approaches, and one of the person who was in the beginning of the TMS mapping in general. And the stage is yours. Please tell us about your experience. I'm very excited about that. Thank you very much.
Thank you very much, Maria, and thank you very much all the Martinos Center to have me here. I'm actually close to Martinos Center in two ways. First of all, I'm from the same place with Thanasis Martinos, so my hometown is the same of your donor. And the other is that I am, as a scientist, I'm original from Biomedical. So Biomedical laboratory and the Martinos Center are somehow, they have a relationship, and that's why also there are many Finnish scientists there. So I'm very proud. I'm going to go directly to my talk. I'm not going to talk about myself, but many of the things are based on my work the last 20 years that I have been doing this TMS and TMS EEG. So let's go. I will talk about how actually neuroimaging can guide TMS. And for some reason, okay. So, and I will start from some practical point of view and why mapping is important, because human brain is complex and human brain functions are unpredictable. You don't know where they are. They cannot be at the same place in the same anatomical, like anatomical landmarks cannot be this, cannot have the same functions. So functionally, we are different. And it can be, we all have in M1 the hand, but it's different from person to person. So this is a very important thing. And it's very important when we talk about tumors and epilepsy, when we talk about operations. So the neurosurgeon has to optimize the extent of resection and to preserve the quality of life and safety of the patient. So we need to take as much more tumor out and keeping the functions in. And this is done by direct cortical stimulation. Usually the neurosurgeons do this on the spot. So here are two examples. So the left example is the extraoperative, which is actually used for not anymore that often for epileptic patients. So there is the surgery, they put a grid of electrodes, and then they close the skull, they pass the cables out, and they do tests for 1 week. And then the other is the typical mapping that they do with for tumors, the intraoperative, as it's called. So it's very important, like they are not, in the world, there are, of course, teams that they can do the extraoperative and the intraoperative mapping very well, but not all. So it's said that at least one mapping method should be used prior to surgeries. So two good ways is MEG and fMRI. And that has been the non-invasive approach, especially for speech mapping, but also for motor mapping and for sensory mapping. So what are the limitations? The invasive mapping, which is the gold standard, is of course restricted to the craniotomy. You cannot map more than that. Then on top, especially for speech mapping, the patient is awake. So this creates many trouble to the neurosurgeon, to the team, to the person itself. The non-invasive mapping has other issues like I don't talk yet about PMS. MEG is temporally specific, fMRI is more spatially specific. Still, they have, like when they are compared to DCS, there are about 20 millimeters of error between the results of the neurosurgeons and the results of these two techniques. So why TMS? So TMS actually, in TMS, in transcranial magnetic stimulation, we induce an electric field to the brain. So it's similar to DCS in a way that it's an electric stimulation. Of course, we don't put the current, we induce the electric field. So it's much weaker, but it's more comparable in a way. The other very important thing with TMS is that we can actually study not only the motor cortex, we can study also behavior. So it has many, many applications. Here I show you a very old picture. We were just trying to reproduce homunculus by just playing around back in 2005. So there's nothing scientific here. Different colors are different muscles and different, of the hand and arm. So in Biomedical laboratory already from 2004, we had this environment of the MEG, also magnetocardiogram. You can see here the first approach of a multilocus, also near infrared spectroscopy. So because we had all these possibilities, when we started doing the motor mapping, we did it together with MEG. So this is a case of a tumor patient. The orange spots are the TMS spots that they gave from hand and arm mapping. So these are the responses based on the electromyograph. And the yellow are from the leg. In between, you see the tumor. The big red spots are the sensory responses from MEG, sensory of hand and of leg. And here you see that how we have been using it. This is also very old. It's of 2007. So you can see here the vein in the MRI, and you can see it also in reality. And then the mapping of the neurosurgeon. So in the beginning, we were able to help them and support them, to show them the landmarks in the brain, to show where they're about, to navigate them. Another example, again, with MEG and TMS is this, it has been published in 2009. We have an epileptic patient, 18 years old girl, and you see the numbers are the grid electrodes, as you see them also here on the right corner. So the red spots are the spots from motor mapping with TMS. The green is the motor knob anatomically. The yellow is the area, the epileptogenic area shown by MEG. And here you see from, this is grid electrodes. So they were stimulating cortically the brain during the week, to find not only the functions, but also the epileptic, the epileptogenic zone. So here we have the red rectangle is the area that they were thinking to, that they were the ictal actually, ictal and interictal discharges, and the white electrodes didn't give any responses. The red ones gave seizures with after discharges, the orange without after discharges. So the purple hand motor response and seizures, so many things. So after all this mapping, we decided to remove the blue perigram, and the result was under it was found a Taylor type dysplasia. There were no seizures, and seizures never came back. No motor deficits, but sensory loss, as it has been actually expected. So it was a surgery with a minimum deficits, actually. And, another very in combination with the other great and another very functional connectivity method, the fMRI, is that here we have a cerebral palsy patient. There was some seizures, and they were thinking of removing the frontal area that they thought that the seizures were coming from. So we did mapping, and we knew already from fMRI that both hands are on the right hemisphere, and that it might be one leg, right leg, but frontally. So we found left hand on the right hemisphere, right hand on the right hemisphere, left leg on where it should be on the right hemisphere, and right leg on the left hemisphere here frontally. So this is the power of mapping, and this is also the power when you have neuroimaging guiding TMS. So this is also another example. We have the distribution of motor functions, and you see within the same subject, like the different hemispheres have different distribution, because this is on musicians actually. So functional neuroanatomy is different for each person. So it's very important. Now, knowing all this, we thought already back in 2008-9 that we should apply TMS for speech. And there was some study of Pasquale Leone showing that you can disturb ongoing speech with RTMS. So we used also the MEG findings from Salmelin and others that they were showing that speech related areas are like showing about 150 to 400 milliseconds after the onset of picture. So what we did this paradigm. And now I show you how it was. And first we were showing objects. These are not, they were not normalized, but still. So objects and and you have to name them, and then you have to name them also during the TMS. So we have been in this particular experiment, we were giving the pulses 300 milliseconds at onset of the picture. There is something, the coil moves away, speech is coming back, you put it back to the correct spot, anomia again. So this paradigm has been, very important and very easy to use. You can put whatever kind of tasks you want, and you can extend to other cognitive functions, not only to speech. So, and We started from that. That was the original design. So we had a camera in front of the cloned images in order to be able to see where is the coil, what is the picture, and how the subject responds. So, and this became a product, an FDA actually approved method. And Nextim has been a spin-off of Biomedical Laboratory. So there are some historical reasons why this went there. So here is another example. Again, we have the extraoperative results from the grid of electrodes. Red circles are electrodes that evoked naming arrest. The green showed motor responses, of the tank is here. And pink was more semantic like errors that you couldn't repeat a sentence. And the same subject with TMS. The red are anomias. The yellow are semantic errors, like semantic paraphasias or phonological paraphasias. And the white are performance errors. So the video allows us to find many, many different errors. So now the situation is that the technique is used all around the world. It has been CE marked and FDA approved. And but still, we need to do a lot. And although it is a field. So the advantage is mapping with TMS is that you can stimulate all possible related areas, left and right hemispheres. You can do basic studies. You can do whatever you want, actually. So this is a big advantage. But there are also limitations. We have false positives, and we have also non-responders. And the other very big problem is that it's user-dependent, like TMS-EEG is very much user-dependent, and speech mapping is very much user-dependent. So what we are doing right now, and we are collecting data actually with very promising results, we want to use MEG and fMRI to guide TMS. Also, we want to use tractography, like diffusion-based MRI, and we do it in a real-time way. In addition, we record the EEG because we want to see the effects on connectivity. But still, we are in a, we cannot use it, but with the idea of using EEG for real-time also speech mapping.
Pantelis, sorry, we have a small technical issue. Let me just... Pantelis? Yeah. Sorry, yes. Let me just make a very short technical break, like for 1 minute, because we have a bit of a problem with internet connection.
Okay.
And you also said that we will have a small break for questions. Just let us know, please, when it's ready.
After I finish with the speech in 5 minutes.
Okay. Great, thank you. Please, meanwhile, just write your questions if you have.
Yours need to be muted. Maybe no sound.
And I could, like, in principle, if there are questions now, I could answer now. So we.
Yeah, if we have anything to address now, please raise your hand. So, again, we have them working, hopefully working microphone over there. Pantelis, could you please check if we can, we don't see him.
You don't see me.
No. Okay, now we can see you beautifully. Okay. So hopefully now it's working. Thank you so much. Please proceed. Thank you, Pantelis.
Shall I continue?
Yes, thank you. Now we can hear you. Beautiful.
Okay. So what we do? We get the object naming with the same pictures in image, the source localization with the MNE, and this is how it looks for different people, different responses. So in this particular subject, the speech network starts getting activated at about 150 milliseconds, and there is 180 milliseconds. There are a lot of, you can see here the timing, a lot of strong responses, and this goes. So we did this was our first pilot. We did a comparison. We usually use 300 milliseconds, which is the default, and that gave very few errors. So it gave about 4 errors out of 120 subject images, trains. So for this particular subject. So then we use the other default timing, which is the 200 milliseconds. So if we don't find a lot of errors with 300, we go to 200, and we found 9 out of 107. And then we used the 180 milliseconds, that's as it's shown in the map that I showed you, the video that I showed you before, and we got 11 errors out of 87, so a bit more sensitive. So this was a good reason to start this project, and it seems that MEG can help a lot with the time. The time is specific for different areas, different time, and here is, if you are interested to look the analysis that we do, and how we do it in practice. So again, now another subject here. It's a bit different, the times. You see the time is a bit later. It doesn't start at 150, it starts about 200. So people have different, like they are different. So we are trying to personalize the mapping even more. And how we do it, we overlay the MNE results into the 3D model of the TMS, and we map the typical areas plus the areas that the MEG highlights. And here is for 12 milliseconds. This is the maps for 340, and we do our mapping. It seems that time plays a big role. So what else we can do? We would like to know how these areas are also structurally connected. So we have developed, Baranay Dogan, Victor Souza, and me have developed this, and others, of course, this method that we give them the seed for the diffusion. So we can have the coil. This is now in your navigator, this in Vesalius, which is open source. So we can put the coil and see how it is connected, the coil over M1, showing the connections to other areas and also to the corticospinal path. So if you move now to DLPFC, it can show you also how it is connected to other areas. Actually, through the Aslan tract, it can be also connected to the speech network, this particular spot. So this is how we nowadays try to block area, for example. So you move the coil and you see how it's connected, so you can follow the lines. So an example, now I use it also like in a computer, in a separate computer, because I cannot put it in the commercial speaking system yet. So do is the so and I can see the connection. This is a case, and this is a need actually. We are trying to identify whether pre-SMA is involved in speech, and it is involved because it is very near the Aslan tract. So sometimes the neurosurgeons, when they remove tumors from there, they get speech impairment. So here I show it. Of course, it comes back the cursor in the middle of the coil. We are in the spot that is connected to the speech network. So we cannot really find it. This, for example, that is connected only to the other side. By moving a bit, we found in this particular spot, and you can see how nicely it's connected. And what we did, we stimulated these areas, and here you see actually that we have got this in this particular subject, anomia, where we showed the performance errors, many different errors. So it will show you here. So all these are evaluation. We have still a lot of work to do, but they are very, very, very promising results. Trying to use this to guide. So before I move TMS-EEG part. I don't hear anyone.
Pantelis, we have a bit of we changed the computer, but we have a bit of internet connection. Could we please try to turn off your video and see whether it might help or not?
Yeah, definitely.
Thank you. Could you try to proceed and we will check how is the sound?
Okay, I proceed then. So TMS-EEG. So when with the EMG and with behavioral responses, we can map very specific things. With the EMG, we can map we can also visual cortex by inducing phosphines. And we can have, as you saw, the disturbance of ongoing speech. However, there are many areas that they don't have these characteristics, but still they are very important, like the frontal areas, the areas involved in the depression network, in all these very important like areas that they don't have the, as I said, the motor or behavioral responses. So TMS-EEG can be a very, very good marker of cortical excitability. It allows us to study reactivity and connectivity. And it can help us even personalize the dose. And it has shown promises in as a biomarker for neurological and psychiatric disorder, especially for MDD patients and for their symptoms. It seems that it can be used. So one thing we know is that TMS EEG is reproducible. Like if we do the experiment today and after 1 week, if we have navigation, if we stimulate the exact same spot, if it's reproducible. So it is reproducible, but it's also very, very sensitive. So the good colleagues from Milan they have been pioneering also in mapping with TMS EEG prior to recording, and I will explain a bit later. They have shown also that very small changes of the coil can result into different signal. And they haven't only showed that, they have showed also that if you induce the same electric field, let's say 120 volts per meter, this is the work of Mario Rosanova, and the previous was the work of Silvia Casarotto. So if you use the same electric field, you induce it, you get different reactivity. So you see in visual cortex, you get much bigger response. And this is in parietal, smaller response, in frontal, even smaller in pre-SMA. But not only different reactivity, you get also the responses oscillate, they ring, they respond in a frequency that it's predetermined by the area. So this is very, very important because you can actually recognize where you are. What we do nowadays, we are involved in a very big grant and a very big study for biomarkers of anhedonia. So we use first the connect comparcellations, and then we use the personalized fMRI data. We get the resting state fMRI to find areas, cortical areas, that they are anti-correlated with area 24. And which is the area where depression is treated with surgery. Usually there are put the electrodes. So what we do this, and then, so these are the areas that we actually map and record. Dorsolateral prefrontal cortex, pre-SMA, dorsomedial prefrontal cortex, intraparietal cortex. So we use a real-time visualization software so that prior to record, we go around, and we have an algorithm that we use it also automatically, and I will show a bit further on, so that if we have a good signal bigger than 6 microvolts and no artifacts, we record. If we have artifacts, we rotate the coil. There is a procedure, nevertheless. And what we do, we go around. Now here you see the parcellation, not the fMRI spots. So we map and we try to find a spot that will give us a good clean signal prior recording with minimum. And this usually looks like this. In DLPFC, things are not that clean as in other areas. Still, you can get, as you see here, a quite good response at 20 milliseconds. So we can get about 6 to 10 microvolts also from here and clean. Not very easily, but still we can. This is another spot, and then we do the mapping. So we find the best one and we say, okay, we will record from here. And we give, in this particular setup, 300 pulses, and we compare it to a fixed location. Fixed location can be either the 5 centimeter rule or the BMF3, which is actually an excellent way if you don't have navigation. Still, because we control prior to recording, we can get much bigger responses and much cleaner responses. And this we hypothesize that will give a bigger predictive power for biomarkers. So here is another example from pre-SMA. You can see again, there is no processed signal. This is a photo from them giving 20 pulses and averaging signals. Like we are already above motor threshold. So the motor threshold is relevant, to be honest, in areas outside the motor cortex. So we have to find a way to quantify and the response and say, okay, if it's bigger than 6 microvolt, that's my threshold or something like this. So here you see that I have a big response, a bigger response, a much bigger response. This is about 15 microvolts at 150% of the motor threshold. So actually we can use TMS-EEG to dose something like a threshold, but based on EEG. And okay, that's another thing, just to show to you how we use real-time tractography. So this is dorsomedial prefrontal cortex, trying to find a good spot. We have a bit of artifacts, not very big. And we stimulate between F1 and F2. So the responses fade out to the other side, but in this particular case, not really. And that's a healthy volunteer. So what we saw actually that we were stimulating here was the red dot. And we saw that the streamlines go exactly towards the direction where F2 and F4 electrodes are. So this is very promising. We also try to evaluate and control for all these findings. And why we do all these things? Because we are preparing the future. So we have the multilocus TMS that we can actually shift electronically the location, like by combining different currents on these two. You see the two different coils. There is one figure of 8 and one ring. So if you combine this and you feed the coils with different currents, you will be able to shift electronically the coil like this. And that was applied in M1. And you can see, like, by moving from minus 15 millimeters from the motor knob to 15 more to the other side of the motor knob, you get at some point MEP responses, high responses, and then they disappear again. So we can create automatic algorithms to find hotspots. And if we combine two figure of 8 coils, we can rotate the electrodes. We can rotate the current of the TMS. So like this. And again, you can optimize your MEP response. But what we did, based on the work of Casarotto and the Milan group and Massimini, we mapped first for a good location without artifacts, and then we automatically rotated the induced electric field. Then we developed a Bayesian algorithm to find the optimal orientation for this particular spot. So in the future, we have already a 5-channel coil that we can rotate the coil, but also rotate the induced electric field, but also translate it at the same time. And this is the way that we do it. We put the coils the one over the other, and you can map actually automatically the cortex. And it will be something like this in the future. So we perturb, we measure, we get the EEG feedback, we have an algorithmic adjustment, and we continue. And now it looks like this.
Go.
So because it's very heavy, we have a robot. We can place it over there. We can give the coordinates in the neural navigator. We can place the coil where we want. If the subject moves, it can be corrected.
Rest back. Yeah.
So when it becomes green,
Rest and
when it
stay.
stays immobilized the subject, then we allow the coil to trigger. So I said everything. So yes, we are many, and there are many different projects involved, and many people together, and we have this major grant, the Connect to Brain for the Multilocus. And these are all the people that have been doing a lot of work. So that's it from me.